Well, hello everybody and welcome back. This is going to be lecture 3, which is essentially the second half of chapter 2. In the most recent version of the Alberts et al. Text, we're going to be going over pages 54 to 68, talking about cellular macromolecules. So essentially that is going to take up most of our time today. Oh, and I did want to say, I'm going to apologize in advance for that black box in the upper right-hand corner. I can't seem to figure out how to get rid of that. So if it's blocking parts of the screen that you want to see, I just want to remind you that you can download the slide decks off Kodiak, and you can always see what's on every slide and use that to kind of fill in any gaps. Trying to keep that upper corner as free of anything as I can, and I'll keep doing research to see if I can get rid of that black box. But that said, we're going to go over macromolecules, which essentially boil down to carbohydrates or sugars as our first class. Lipids generally, but we're going to focus most of our attention on fatty acids, a subgroup of lipids. The amino acids, which are, of course, the building blocks of proteins. Nucleotides, which are the building blocks of nucleic acids, RNA, and DNA, and those are the 4 groups of macromolecules. Along the way, we'll also be defining and talking about monomers, polymers, the importance of sequence, and then we'll wrap up talking about non-covalent bonds that hold these macromolecules together, echoing back from some of the material that we've already talked about when we were talking about chemistry and bonds earlier. So, now that we know that life is essentially nothing more than an applied chemical system, and we have a sense for the different ways that atoms can come together to form molecules through covalent bonds or salts through ionic bonds, we have to wonder, well, what comes next? Just making molecules doesn't make life possible. So what is it about the molecules that are in a cell that make life possible? And maybe even more importantly, what do those molecules do in a cell? A cell is formed of carbon-containing compounds. Organic chemistry is called organic because it is carbon-containing molecules that are responsible for life. And as we just reviewed in the opening slide in the outline, cells contain those 4 major families of small organic molecules. Those are the macromolecules: fats, nucleic acids, sugars, and proteins. Sugars are both an energy source and how we fuel our cells to keep surviving, as well as the subunits for more complex polysaccharides. Those can be involved in energy storage, but they also play a host of other functions that we'll touch on today. Fatty acid chains are the main component of the cellular membrane, so those are important for keeping this hydrophobic barrier around the cell. Amino acids are the subunits of proteins, and proteins, of course, are critically important to life. And as I already mentioned, nucleotides are the subunits of DNA and RNA. So with these 4 ingredients, with these 4 relatively simple organic molecules, we can make all of the molecules that are responsible to keep a cell alive. We can make life possible. So back to that idea of organic compounds, carbon-containing molecules. If you disregard water, so a quick side point about water, there's no doubt at this point that life first evolved in the early primordial oceans of the Earth. Life is an aqueous phenomenon. We talked about that earlier. Water is the universal solvent, and it's clear that life and the molecules that make life first evolved and came to be in the oceans. When that early life eventually went terrestrial, when that first slippery eel-like thing had the courage to climb out of the ocean and start to exist on land, the only way to leverage all of that evolution that had already occurred was for that life to essentially take the ocean with it. If you look at the salinity of the water in our bodies and the salinity of the water that's inside a cell, the salinity of that water is very close to the salinity or salt concentration of ocean water. So very much, it's not a metaphor, it's not an analogy. When life left the oceans, it took the ocean with us, and we're about 70% water. So by volume, by mass, life, including us, is more water than anything else. If you disregard that water, and you look at all the non-water molecules, nearly all of the molecules in any living cell contain carbon. They are organic. And we already know the reason for that, or we should if we think back of the material we've already covered. Carbon has 4 unpaired valence electrons. Carbon is capable of making 4 covalent bonds, and because of that, carbon is the universal Lego piece joiner of life. Atoms can be stuck to other atoms through carbon. Carbon is the backbone atom of life, just because of the versatility of it being able to make 4 covalent bonds. When atoms need to connect to other atoms in 3-dimensional space, carbon is the best way to connect them, and that's why life is built on carbon, on this planet at least. Carbon's also involved as one of the central atoms for many what we refer to as functional groups. Functional groups are relatively small, meaning they contain a limited number of atoms, small combinations of atoms that we find in a lot of different biomolecules. And they essentially serve the same purpose or very similar purpose in all of the macromolecules that they're part of. So some examples of a functional group. A methyl group is a functional group. It's about as simple as you can get. A methyl group is 1 carbon and 3 hydrogens, and we find methyl groups on DNA, we find methyl groups on protein, methyl groups are used for signaling, methyl groups are used for modifying, so it's an example of a functional group. You have here a fatty acid. A fatty acid is a functional group. We typically refer to it as an acyl group in that context, but a very long chain of carbons and hydrogens, a hydrocarbon, is a functional group. We've got alcohols, aldehydes, ketones, carboxyl groups. In this case, it's a carboxylic acid because it still has its proton, but that's essentially a carboxyl group. And then we can have esters. Esters is when we have a combination of an acid and an alcohol. These are all carbon containing. We see carbon serving as that bridging molecule, that erector set piece in the center of all these functional groups. These are functional groups. Well, those are functional groups that contain carbon and oxygen, but we can have functional groups that contain carbon and nitrogen, things like amines, amides, as well as other functional groups that contain sulfur, such as a sulfhydryl group, and even phosphate groups are functional groups, and they don't contain carbon directly for a phosphate group, but they are bridged to other molecules through carbon, because again, carbon makes up the backbone of so many biomolecules. So we've got carbon serving as the linker of the atoms that we need. And now we have, I guess you could say, like prefabbed, right? Prefabricated collections of atoms that do a similar function no matter what molecule they're part of. And those are our functional groups. Now on to the 4 families of macromolecules. Should be no surprise now, but we're talking about complex carbohydrates, lipids and fats, proteins, and nucleic acids. And each one of those, everything you see listed on the right, is a polymer made up of small subunit monomers that are linked together to create a larger structure. The monomers of polysaccharides are monosaccharides, or simple sugars. The monomers of fats and membrane lipids are fatty acids. The monomers of proteins are amino acids, and the monomers of DNA or RNA are nucleotides. Nucleotides are linked together to create a large nucleic acid. Amino acids are linked together to create a large protein, and so on. This gets at the idea of anabolism and catabolism, ideas that we will return to again and again, I think for the next 4 weeks. We'll keep coming back to this. But to introduce it here, anabolism is the making side of metabolism. When we take monomers from the foods we've consumed and energy yielded from the foods we consumed, and we use that energy to string those monomers together into the biomolecules we need, we are doing anabolism. When we consume complex polymers from living things that we have eaten, and we break those polymers down into their individual constituent parts, into their individual monomers, and in doing so, we break covalent bonds and release the potential energy from those covalent bonds, we are doing catabolism. So catabolism is the breaking down of foods to get those monomers and that energy released, and the anabolism is then using those monomers and that energy to make the polymers that we need. And together, anabolism and catabolism make up metabolism, the combined sides of that coin. So sugars, let's talk a little bit more about sugars. Sugars can be used both as an energy source as well as monomers for more complex polymers that we need in the cells that are sugar-based. And we'll focus on the energy role of sugars later on this semester. We're going to spend more time today just talking about sugars generally and how they work as polymers. A single simple sugar monomer, the simplest sugars that are available in life, are called monosaccharides. Mono for one, and saccharides as the suffix for sugar. And they are carbohydrates. So let's dissect that term, carbohydrate. I don't want to gloss over that because there's so much meaning there. When we say carbohydrate, when we use that term, what we really mean is a hydrated carbon. Carbohydrate is a molecule that contains carbons that have been hydrated. Sugars are really nothing more than chains of carbon stuck together in a chain with a water molecule put on each one. Indeed, the general formula of all sugars shows us that. The general formula of any sugar is CH2ON, where that N simply refers to the total number of carbons in the molecule. In other words, the total number of hydrated carbons in the molecule. Because look, here's water stuck to one carbon, and if this N was 6, as it is in the case for glucose, right? Glucose is C6H12O6. Well, then we have 6 carbons with 6 water molecules, 1 water molecule on each one. Sugars are nothing more than hydrated carbons, carbons in a chain with water on each carbon. However, that's not all a sugar is. Each individual unique sugar molecule is not only defined by the number of carbons and waters that it has as part of it, it's also defined by its three-dimensional shape. The orientation of the atoms of the sugar molecule and how they line up in three-dimensional space is what gives the sugar its identity. Two different sugars can have the same exact chemical formula, but they can be completely different sugars with different chemistries and different roles in the cell based on nothing other than their shapes. And this gets us to the idea of isomers. Isomers, I'm assuming, is a concept you've learned about already in your chemistry classes, but the idea of chemically identical molecules in the sense of the number of atoms they contain, but because of the shape of those molecules, they have very different functions. Spearmint and rye is a good example. The molecules that give us the spearmint taste and the molecules that give us the rye taste have the same exact number and types of atoms. The reason why they taste and smell differently to us is simply because of the orientation of those atoms in three-dimensional space. So what you see here is glucose, galactose, and fructose. Each one of these sugars contains 6 carbons. Each one of these sugars is a carbohydrate. And so you can see each carbon has a water on it. Each carbon has a water on it. We usually have a double bonded carbon somewhere in a sugar, but all the other carbons have been hydrated. If you count up all the carbons, all the oxygens, and all the hydrogens in all 3 of these sugars, they are exactly the same. The chemical formula for glucose, galactose, and fructose is C6H12O6. However, these are 3 completely different sugars. The reason why is because of the position of the double bonded oxygen. Is it an aldehyde, as we see here, the double bonded oxygen is on the end, or is it a ketone, as we see here, where the double bonded oxygen is in the middle? And also, what is the position of the hydroxyl groups and the hydrogens? Here we see the only difference between glucose and galactose is the orientation of the hydroxyl and the proton on the 4th carbon. These are all isomers of each other. These sugars have different functions in the cell. They have different chemistries. They are identical in their chemical formula, but different in their shape. Sugars can not only be linear, but they can also be ring-shaped. These are the rings of glucose, galactose, and fructose. And again, we see those spatial differences. Where these functional groups fall on the sugar of the ring is different for each one of these isomers. In glucose and galactose, because of the position of the hydroxyl group, we see for one, the hydroxyl group is pointed down away from the ring, and in the other, the hydroxyl group is pointed up. And because of the position of the double bonded oxygen, we see that this region is in a different place for fructose than it is for galactose and for glucose. And then I'm not going to get too much into boat and chair, the different forms or orientations of sugar rings, but you can have different three-dimensional orientations of sugar rings. How these atoms fold in three-dimensional space relative to one another in the ring can also be different for different sugars. And so sugars have an enormous diversity of chemistry because of these differences, not only in the number of hydrated carbons, but also is it linear or is it a ring, and what's the orientation of the functional groups in that molecule, both in the linear and the ring form. All of these things combine to make sugars unique from one another. Each macromolecule, for sugar or any other polymer, each macromolecule is a polymer of monomers held together by a unique and named bond. The bond, the covalent bond that holds monosaccharides together into a polysaccharide, is called a glycosidic bond. The covalent bond that holds amino acids together, as we'll see next week, is a peptide bond. The covalent bond that holds nucleotides together is a phosphodiester bond. Each one of these polymers has a uniquely named covalent bond that holds the monomers together into a chain. And again, for sugars, the name of that bond is a glycosidic bond. Two monosaccharides can be linked together by a glycosidic bond, giving us a disaccharide, di for two. And here's that reaction occurring. We've got alpha glucose and beta fructose coming together. We want to link them together to make them a disaccharide. We're going to steal a proton from this hydroxyl group and rip off the entire hydroxyl group here, giving us H2, those two protons, plus the oxygen, H2O. And so water is going to leave as a leaving group. This one remaining oxygen that we see here is then going to form a covalent bond with the carbon here, acting as a bridge. And that oxygen is going to bridge those two sugars together, giving us our disaccharide. And this is now a polymer by definition. It is more than one monomer held together. And this right here is our glycosidic bond. You can add another sugar onto this chain and make a trisaccharide if you'd like. If you do a fourth sugar, we usually move away from the specific prefixes and then we just use the term oligosaccharide. Really, any saccharide that has between 3 and 10 monomers is going to be an oligosaccharide. And then if we continue adding sugars to make a very large and complex sugar, then we're going to use the term polysaccharide. Polysaccharide is going to refer to any complex carbohydrate that has more than 10 monomers. And what you're looking at here on the screen is glycogen. Glycogen is how we store excess sugar for the future, during fasting, during sleeping. And each blue hexagon that you see there in that diagram is actually a single monomer of glucose. So there's many, many, many glucoses linked together here on this glycogen tree, simply for storage, for energy storage. As I've already alluded to, the reaction type that makes these polymers is referred to as a condensation reaction. Just like a cool glass of cold water on a hot, humid day will exhibit condensation. Condensation on the outside of that glass is the drawing of water out of the air, and that water condenses on the glass. So condensation is the removal of water. In this condensation reaction, water is removed. As I showed you before, and I'll show you again in a second, if we have two hydroxyl groups, as is the case for sugars, if we have two hydroxyl groups attacking one another, and one hydroxyl group leaves completely, and the proton of the other hydroxyl group leaves as well, those two H's and that O will come together to make water. Just like water grabbing onto that cool glass, water leaves these sugars as a condensation reaction. Sometimes people are referred to this as a dehydration reaction as well. Same logic applies, dehydration, the loss of water, but condensation reaction is more typical and more commonly used, and those are the terms that we'll be using this semester. This diagram is nice because it lets you track the atoms of the water. Those are the atoms that we see in red. This is the entire hydroxyl that we're losing, and it's this proton that we're losing as well, giving us H2O. Water is our leaving group, and this is the oxygen that's left behind to serve as the bridge of the glycosidic bond. Now, we're going to see when we talk about condensation reactions in amino acids that it's not always two hydroxyl groups that are attacking one another, but all of these bonds that make the polymers of the macromolecules are going to be made through condensation. And we're going to lose water as a leaving group. So the generic kind of representation of any polymer creating condensation reaction is represented here. A in green is one monomer, B in blue is another monomer. Those can be two sugars, two amino acids, two nucleotides. We're going to have at least a proton on one of those monomers and a hydroxyl group on the other, and those three atoms will be lost. They'll leave as a leaving group of water. There's your condensation. And the electrons, the unpaired electrons left behind between A and B, will then pair up, and those electrons will do their nice figure 8 orbiting around these two atoms, joining these monomers together in a covalent bond. Condensation reactions make the bonds that create polymers, and hydrolysis reactions break those bonds. Hydrolysis literally translates to splitting with water, And if it was water coming out of these two monomers that allowed us to join them, then if we force water into that bond, we can break it. And that's indeed what a hydrolysis reaction is. We give a proton back to A, we give a hydroxyl group back to B, and in doing so, we split the shared electrons that were creating that polymer bond, and we break that bond. In the case of sugars, this would be the glycosidic bond that we've made and the glycosidic bond that we've hydrolyzed. So again, condensation, water losing. We're going to see condensation reactions again and again today, and as we continue on in the earlier parts of this semester. Now, for the last few slides, the title at the top has been Sugars are an energy source and monomers. Well, we've definitely hit on the monomer thing, I think. We can string these monosaccharides together to create an oligosaccharide or a polysaccharide. But what's the idea here of the energy forming? Well, hold on to that for a little while. We've got a whole mini unit all about how we get the energy out of sugars and use that energy to make ATP. That's the cellular respiration story that we'll be telling in week 6. But suffice it to say here that part of the reason why polysaccharides even exist is to store energy for later. I've referred to glycogen already. Glycogen is a polysaccharide of glucose that animal cells use for short-term storage. Well, plants do something quite similar. Starch is made up of nothing but glucose. There are 2 different polysaccharides that make up starch, amylose and amylopectin, but the idea is generally the same. You take individual excess glucose monomers and you link them together one by one, making glycosidic bonds via condensation reactions, and you create a large polymer or polysaccharide of glucose. And then, as the sun goes down, or it's a cloudy day and that plant is unable to engage in efficient photosynthesis, the plant can survive for another day by breaking down its starch stores and feed on the sugar that it's stored for just that occasion. So polysaccharides, but for energy, as an energy source. That said, sugars are also purely structural. Cellulose, which is the component of the plant cell wall, all true plants on this planet are standing upright and have the durability of their stems, of their bark, of their outer surface because of their plant cell walls. And those plant cell walls are made of cellulose, and cellulose is nothing more than structural glucose. Glucose chains that are linked together both covalently and through hydrogen bonds to create this very durable watertight wall around each individual plant cell. So nothing more than a polymer of glucose, not for energy storage, but for structure. Chitin. Chitin is also a polysaccharide. This is the component of insect exoskeletons. It's a polymer of N-acetylglucosamine. Glucosamine is what it sounds like. It's glucose with an amine group on it. And so it's a nitrogen-containing sugar. But we take these glucosamine monomers, we link them together by dehydration reactions, by condensation reactions, and we can create an armor, this wall, this crunchy, durable, watertight wall that safeguards insects. Even slime and mucus, things like mucus that protects our airways, the slime of a crawling snail. These are polymers of sugar. They're so sugary, if you will, that they attract water to them. It's almost like a thick, viscous water and sugar amalgam or combination, but they are sugar-based. The non-water molecule in slime and mucus is a polymer of sugars. And oligosaccharides are often linked directly to proteins, giving us these hybrid molecules, which we call glycoproteins. Glyco for sugar and proteins, of course, for protein. And glycoproteins are responsible for an entire host of multicellular activities. In fact, the whole idea of blood type is based on glycoproteins. I have O-type blood. Well, that means my red blood cells don't display sugars on their surface, or at least not the sugars of the AB typing system. The O really stands for 0, for nothing. Individuals with A-type blood, well, their red blood cells display one kind of sugar, sugars that are linked directly to the proteins in the membrane of those red blood cells. Individuals with B-type blood, they display a different kind of sugar. And AB blood means you display both of those types of sugars. So our red blood cells display sugars on their surface, sugars that are covalently linked to proteins embedded in their membrane. In fact, the way that a fertilized egg finds and implants itself into the uterine wall is through an interaction between proteins and glycoproteins. It's a sugar protein that allows an embryo to embed into the uterine wall and begin making a placenta. It is these sugar-based proteins that allow a white blood cell to recognize a site of infection or a lesion. So these glycoproteins are critically important to our immune system, to the perpetuates of our species, to how our blood behaves, to how we recognize self and how we recognize foreign, all based on these glycoproteins. So sugars, I mean, come on, right? Structural, energy sources, highly complex in their three-dimensional isomers, in their chemistries, and then critically important not only to our well-being, but also to our species continuing. Moving on to our second macromolecule class, lipids, but again, I'm going to restrict our conversation today to fatty acids. Fatty acids are not carbohydrates. Fatty acids are hydrocarbons. What are the only two words you see there? Hydrocarbon, hydrogen and carbon. That's all a fatty acid tail is, carbons and hydrogens. Look at that. That's a long chain of carbons, just like a sugar, but there's no water on those carbons. There's just hydrogen on those carbons. And since nothing you see there is electronegative, since you have no oxygen there at all, it's just carbon and hydrogen, both two very weakly electronegative atoms, we've got no hydrophilicity here. There's no charge. There's no partial charge in that fatty acid tail. There's no full charge in that fatty acid tail. That's completely hydrophobic, and fatty acids are hydrophobic. Without oxygen, we've got no polarity. Without polarity, we don't have partial charges, and we've got hydrophobicity. That said, I'll go back for just a second. That said, every fatty acid is typically capped with a carboxyl group that has lost its proton, that has acted like an acid, so it has a full negative charge. So there is charge at the very tip of a fatty acid tail, and that creates a molecule which is amphipathic. Amphipathic is a word we use for a molecule that has a very large and significant hydrophobic region, and then structurally separate from that, we also have a very strongly hydrophilic region. So this is a molecule that has a foot in each pond. Purely hydrophobic, purely hydrophilic, and those two ends are spatially different and chemically different. Through that charge, that a carboxyl group, we can attach this fatty acid to other molecules and attach other molecules to this fatty acid. So that's our connection point for these molecules. Now, fatty acids can be either saturated or unsaturated. You've probably heard these terms with regards to diet, right? Saturated fatty acids are things like butter, not so healthy for you. Unsaturated fatty acids, things like olive oil, presumably more healthy for us. Well, there's a chemical reason for those terms. When a carbon chain is saturated, what it's saturated with, what it's full of, if you think of a saturated sponge, what it's full of is hydrogens. Saturated fatty acids can fit no more hydrogens on them. Every single carbon of that fatty acid tail has as many hydrogens on it as it can possibly fit. And for that to be possible, and for a fatty acid to be saturated, there can be and there are no double bonds. This gives that saturated fatty acid a very straight linearity. When we have an unsaturated fatty acid, well, that means that we're not saturated with hydrogens. In other words, there are some carbons that could, presumably, take on more hydrogens if forced to, but they haven't. It's like a sponge that's partially full of water. This is a carbon chain that's partially full of hydrogens. The reason why not every carbon is fully hydrogenated is because of the presence of double bonds. So when we have double bonds in a fatty acid tail, we are unsaturated, but also those double bonds give that fatty acid tail a three-dimensional kink. Now, the reason for the health difference is because the saturated fatty acids, because they're so straight, they can be lined up very densely. You can fit a lot of saturated fatty acids into a relatively small space. And because of that higher density, these fatty acids tend to be solid at higher temperatures. Think butter as a solid brick on your counter at room temperature. When you have an unsaturated fatty acid, that kink means you can't really stack these fatty acids as close together because they each take up more three-dimensional space. And because of that, you can't squeeze them as densely together as you might for a saturated fatty acid. Well, lower density means liquid at room temperature, just like olive oil. So fatty acids that will get close and dense together to make butter on your kitchen counter also have the potential to get close together and make that kind of solid consistency in your arteries, making them unhealthy. Something that's very loose and liquidy at room temperature, like an unsaturated fatty acid, is also going to be looser and more liquidy in your bloodstream, leading to better heart health. Yeah. So that's what we mean by saturated and unsaturated fatty acids biologically, but it ties very nicely into things we've encountered in the everyday world. Now, fatty acids very rarely exist in isolation. They're very typically part of other larger polymers, not surprisingly. We're going to link them with reactions to other molecules and have more complex molecules as a result. We're going to talk about two of those molecule types. One is a triacylglyceride and the other is a phospholipid. But what we're looking at here at the bottom right is a triacylglyceride. I'll give you more details on that in just a second. But what I want to emphasize here is that the triacylglyceride on the left is made up of nothing but saturated fatty acids. Look how straight those fatty acids are. Those fatty acids are dense. They're going to be more solid and less viscous in our arteries and therefore more dangerous. The fatty acids on the triacylglyceride on the right, well, they're obviously unsaturated because they got all those kinks in them. All those kinks are taking up more room, a much looser, much less dense triacylglyceride. It's going to flow much better in the bloodstream. It's going to be a lot healthier for us. So fatty acids can be structural. We'll see the role that they play in the cell membrane in just a second, but they're also, much like complex carbohydrates, a source of stored energy. In fact, we can get more energy per unit mass, more energy per gram in fatty acids than we can get in sugars. There's on average about 6 times more potential energy in fatty acids than there are in glucose. And that's because of the nature of the covalent bonds. The covalent bonds of fatty acids they're more stretched, they're longer, there's more strain on those bonds, giving them a higher potential energy. The bonds in glucose and other sugars, they tend to be shorter, more stable bonds, and because they're shorter and more stable, they're less energetic. So this makes fatty acids a very potent energy storage molecule, but fatty acids are very difficult to break down. Sugars, we're going to see in just a couple of weeks, we can break sugars down just like that. But fatty acids, they take a lot of work to get that energy out. So fatty acids are a much better long-term energy storage molecule. So the way I always like to think about this is if I'm going to skip breakfast or I slept for 10 hours, my body is going to use sugar stores like glycogen for those short-term fastings. It's a short-term sugar storage, short-term energy supply. If I skip a meal, I'm going to go into my glycogen stores. My fatty acid stores, my triacylglyceride stores, that's for getting me through the winter as a bear. Right? If my last meal was in late October and my next meal is going to be in early March, it's fats that I want to store that energy in because I've got 6 times more energy per pound. And so that's going to be a long term. So glycogen and complex carbohydrates is a like daily energy storage, and fatty acids are a seasonal energy storage. I've already alluded to it, but fatty acids are usually stored for energy as these triacylglycerols. Ooh, another term. I love these terms. Let's break them down. Tri, well, we all know what that means, 3. Acyl, I already told you that acyl is the prefix we use for fatty acids as a functional group. So when you see acyl, that's fatty acids. That's the functional group of a fatty acid. So triacyl, 3 fatty acids held together and stuck to a glycerol. A glycerol is a 3-carbon molecule, basically a 3-carbon sugar. You can think of it as that. And that is the clothespin, the paperclip, the joiner of our triacyls. So here's 1 fatty acid, that's 1 acyl. 2 fatty acids, diacyl. 3 fatty acids, triacyl. And what's holding those 3 fatty acids together? A glycerin clothespin. So there's the glycerol with 1 carbon of the glycerol holding onto the first fatty acid, carbon number 2 to the second, and carbon number 3 to the third. So glycerol is our carbon-based organic backbone holding these 3 fatty acids together, and the only purpose of triacylglycerols is this long-term seasonal hibernation energy storage. The fat, like fat that we think of as fat, that's triacylglycerol fat, and that's for energy storage. And when energy is needed after a long-term fast or starvation, it's these fatty acids that will be broken off of glycerol, and then broken down to individual carbons and metabolized to release that energy. Similar in structure to triacylglycerols, but quite different in function, are phospholipids. Phospholipids contain 2 fatty acids instead of 3. They have glycerol holding it all together. But in that third slot of glycerol, again, because we don't have the triacyl, we don't have the third fatty acid, in that third slot of the glycerol, instead we have a very hydrophilic, usually ionic group. Phosphates are the most common, that's why it's a phospholipid. And so if we look at a phospholipid, we've got a really unique molecule. Here's the glycerol in green, right? 1, 2, 3 carbons holding everything together. Here's the first fatty acid. Here's the second fatty acid. And I could ask you, like I might on Friday, what kind of fatty acid is this? Give you a second to think about that. Oh, that's a saturated fatty acid. Saturated fatty acid because it's long, it's not kinked, right? And then here's the third carbon of glycerol, but we don't have a third fatty acid there because this is not a triacylglycerol. Instead, on that third carbon, we've got this phosphate group. And that phosphate group is charged, full charge. It's an anion. And that full charge is going to be very, very hydrophilic. So phospholipids are extremely amphipathic. We've got this hydrophobic region here made up of the fatty acids and a very hydrophilic region here made up of the phosphate. And because they're amphipathic, it's phospholipids that are most idealized and evolved to create membranes. If you drop a whole cluster of phospholipids together into water, all of those hydrophobic tails are going to be shunned by water, and they're going to be pushed together as hydrophobic, and I'm doing air quotes, you can't see me, but I'm doing air quotes. They're going to be pushed together as hydrophobic interactions. The enemy of my enemy is my friend. All of these hydrophobic tails are the enemy of water, and water is driving them together to increase entropy. However, water's got no problem at all with the phosphate groups. Water loves interacting with those negative charges. Specifically, the partially positive hydrogens of water will love interacting with that full negative charge of those phosphate groups. And so these phospholipids will immediately, spontaneously, and requiring no energy, form either small micelles, these solid balls of fatty acids, of phospholipids, where the fatty acid tails are in the center and the hydrophilic head groups are on the outside. But much more commonly, they will form a bilayer, where the hydrophobic tails are all in the center, and we've got 2 layers of hydrophilic head groups on either side of the bilayer. That's what a cell membrane is. And I know I'm saying a lot, not showing a lot of pictures. We've got an entire week dedicated to cellular membranes, and we'll go into all of this material in detail then. Oh, but look, here is a picture. So this is a stylized version of this diagram here. I love the textbook in so many ways, but what I really love about the textbook is how consistent it tries to be. Here we've got a little bit more of a detailed representation, the fatty acid in orange. We've got 2 saturated fatty acids here. Here it looks like we've got 1 saturated and 1 unsaturated. Here's the glycerol in green. Well, there's the glycerol in green. Phosphate group here in yellow, phosphate group. And then attached to the phosphate group, we usually have more polarity, molecules that contain either oxygen or nitrogen, giving us more charge. So this is very, very hydrophilic, and this here is very, very hydrophobic. And just as I described a moment ago, in the presence of water, water is going to drive all those hydrophobic tails together into the center, where water is completely excluded. These are the hydrophobic interactions. And water is interacting just fine with the hydrophilic head groups. And this is the barrier of the cell membrane. Things cannot easily pass through this barrier because in order to be in the water in the first place, you're hydrophilic, and then hydrophilic things are shunned by hydrophobic things. So this gives us a nice kind of waxy, oily seal around the perimeter of the cell. So when we talk about the semi-permeable lipid bilayer, this is what we're talking about. We're talking about this, I don't want to say polymer because these phospholipids are not held together by covalent bonds, but we're talking about this complex structure made up of many, many phospholipids that are driven together to create a membrane due to the mutual exclusion of the fatty acid tails by water and water's favorable interactions with the hydrophilic heads. Okay, so now we got sugars and fatty acids under our belt, moving right along to amino acids. Amino acids are the subunits of proteins. Proteins are polymers, and amino acids are the monomers that make those polymers. And I'm going to level with you all right now. I'm going to make a personal admission that brings me an enormous amount of pain. I'm a molecular geneticist. That's what my PhD is in. I studied DNA for my entire PhD. My 2 postdocs were also based on DNA and gene regulation. I love DNA. I'm fascinated with it, and I've dedicated my entire professional life to DNA. That said, I gotta be honest with you all. It's really the proteins that do everything. Proteins do everything, right? And we've already talked about this. Proteins are involved in signaling. Proteins are involved in metabolism. Proteins are involved in catalysis. There are structural proteins. There are receptor proteins. Everything that is making life possible is the work of proteins. If life is a mechanical process, and it is, then the proteins are the machines that are making that mechanical process possible. And proteins are nothing more than polymers of individual amino acid monomers linked together by, here it is, condensation reactions. And the bond that holds those amino acids together is a peptide bond. It was a glycosidic bond when we were talking about sugars, and it's a peptide bond here. And here is that reaction. We have an amino acid on the left. We have an amino acid on the right. Just as I've warned you, it doesn't always have to be 2 hydroxyl groups for this. In this case, we're going to strip off this entire hydroxyl group and just this 1 proton, leaving an unpaired electron on this carbon and an unpaired electron on this nitrogen. And then those 2 unpaired electrons are going to pair up, and they're going to orbit around the carbon and the nitrogen, giving us a covalent bond, giving us that peptide bond right here. And that peptide bond is going to hold those 2 amino acids together into a dipeptide, and we can continue that process and string additional amino acids together. But let's not forget, this was a condensation reaction. Here's water as our leaving group. This hydroxyl group and this proton came together to make water, and we have made this bond by removing water, and we can break this bond by hydrolysis by adding that water back. So if proteins are so important, then we've got to talk about proteins considerably in this class, and we will. We're going to open the door to proteins here in the next few slides, and then all of next week is dedicated to proteins. Proteins is going to take up the entire summation of what we discuss next week. All amino acids, not surprisingly, are organic. They're built around a carbon skeleton, and every amino acid has a central carbon at its center, referred to as the alpha carbon. And that alpha carbon is linked to a proton that takes up one of its unpaired electrons, a carboxyl group that takes up another one of its electrons, an amino group, which is why we call it an amino acid, and then the last unpaired electron is given to a variable group. This group is called the R group or the variable group because each of the 20 amino acids has a different functional group down here. So every amino acid has an alpha carbon with an amino group, a proton, and a carboxyl group. But what's in this 4th position here is what makes each amino acid different. Amino acids, again, are the monomers of proteins. We make these peptide bonds via condensation reactions between the carboxyl group of one amino acid and the amino group of the next. So if this is amino acid number 1 here, phenylalanine with its big hydrophobic ring, we'll get to that next week. Here's the carboxyl group of phenylalanine. Here's the amino group of serine, and we've made a peptide bond between those 2. Here's the carboxyl group of serine and the amino group of glutamate, and we've made a peptide bond between those 2. Here's the carboxyl group of glutamate and the amino group of lysine, and we've made a peptide bond between those 2. The peptide bonds are joining the individual amino acids together into a polymer, and they are always between the carboxyl group of the first amino acid and the amino group of the next. All made through condensation reactions, all made by the loss of water. This implies that proteins have directionality, and indeed they do. Proteins have an established beginning and end. The beginning of any protein sequence, the beginning of any chain of amino acids, is its N or amino terminal end. This is the beginning of this protein sequence. This protein sequence reads phenylalanine, serine, glutamate, lysine, and it does not read lysine, glutamate, serine, phenylalanine. We go from N to C, from amino to carboxy. A long chain of amino acids is a polypeptide, poly because it's a polymer and peptide because it's a protein. And then once that long chain of amino acids is created, that amino acid chain is going to fold into a 3-dimensional shape, and we'll talk all about that next week. You might remember from previous classes, this is the idea of primary, secondary, tertiary, and quaternary, but we'll cover all that next week. And then once this polypeptide has that three-dimensional structure, that three-dimensional structure gives that polypeptide its, wait for it, its function. Structure equals function. We're going to come back to this concept more times than any other concept in this course, as is the case for cells, as is the case for sugars. For proteins, the structure of the protein gives it its function. Panel 2.6 in the textbook, I really wish I had written down the pages, and I don't have the textbook near me right now, but panel 2.6 in the textbook does a wonderful job of simply and straightforwardly showing a lot of the concepts that we've already talked about and some that we'll talk about in just a second. I definitely recommend this as a study resource, and I think that it could be very helpful as you begin to prepare for this week's quiz. So to recap some of the things that's in that panel, there are 4 families of amino acids: acidic, basic, uncharged polar, and then completely nonpolar or hydrophobic. We're going to recap a lot of this information next week, but just to scratch the surface here, the acidic side chains are negatively charged. You can think of it as an acid that's already donated its proton, and so there's a negative side chain remaining. The basic side chains are fully positively charged, they're cations, and so obviously the acidic and the basic side chains are very, very hydrophilic because they're fully charged. Our uncharged but polar side chains are what they sound like. There's no full charges here. They're not ionic, but they are polar because they contain nitrogen and or oxygen, our electronegative atoms. And because of that electronegativity, we have polar bonds, which means we have partial charges, which means water can interact with these side chains just fine. Things like asparagine, glutamine, serine, threonine, tyrosine, these are our uncharged but still polar amino acids because they contain these hydroxyl groups or these nitrogen-containing groups. And then we have the nonpolar side chains, like no charge at all, no full charge, no partial charge, really nothing but hydrocarbons if you look at these. And these nonpolar side chains are going to be hydrophobic. Water will shun them. These amino acids will be driven to the interior of a folded protein. They are hydrophobic. And so between the 20 amino acids, it's really the different chemistries and shapes of these side chains. By chemistries in this context, I really just mean charge, full charge or partial charge. And the shape, is it big and bulky? Is it small and nimble? The different chemistries of these side chains and the sequence that the side chains are put in the context of the chain of the amino acids is what allows the protein to fold into the shape that it takes and then ultimately have the function that it does. And I didn't want to leave this panel and this material without pointing out that just like we have isomers for the sugars, we have isomers for amino acids as well. These are non-superimposable mirror image isomers or stereoisomers. We have an L form and a D form is the way that we refer to them for amino acids. And interestingly, suspiciously, one might say, there are only L form amino acids found in living cells. We can find D form amino acids out there in nature, but no living cell on Earth uses D form amino acids, only L form. Why that's the case, there are some theories about, but no solid footing there. So just an interesting little tidbit. Wouldn't it be amazing if we do find life somewhere else on some other planet and it's built nearly identical to us, but they use D form amino acids? So basically, it would be the molecular mirror image of life on Earth. There's nothing stopping that from being so biochemically. It appears to be largely happenstance that life evolved to only use L here. And again, I'm going to leave it at that for the proteins because we're going to spend a whole week on them next week. But moving on to the nucleotides. Nucleotides are the subunits of the nucleic acids. That's deoxyribonucleic acid and ribonucleic acid respectively. These are polymers that are made up of nucleotide monomers strung together by, in this case, phosphodiester bonds. So that's the name of the covalent bond that's going to make the polymers of nucleic acids. Phosphodiester bonds for nucleic acids, peptide bonds for proteins, glycosidic bonds for sugars. And, not surprisingly, starting to become a theme, these bonds are made by condensation reactions. Water is going to be removed. Every nucleotide monomer consists of a nitrogen-containing ring or rings. That's the nitrogenous base. This is the adenine, guanine, cytosine, thymine, uracil that you're probably already familiar with. And that nitrogen-containing base is linked to a 5-carbon ribose sugar. That 5-carbon ribose sugar is also linked to at least one phosphate group. Now we can have 2 or 3 phosphates as well, but at least one phosphate group. So if you have a ribose sugar linked to a nitrogenous base and also linked to at least one phosphate, you've got yourself a nucleotide. Most of the monomers that are involved in making nucleic acids are triphosphates. They contain 3 phosphates in a row, and the importance of that we'll get to near the end of the material here today. If the ribose sugar has 2 hydroxyl groups at its base, as we see here, one hydroxyl group coming off the 2' carbon and one hydroxyl group coming off of the 3' carbon, then we're looking at ribonucleic acid. This is a ribonucleotide. It's going to be a subunit of ribonucleic acid. It's going to be part of RNA. If the ribose sugar has been deoxified, that's my own made-up word, but I think it gets the point across. If an oxygen has been removed from the 2' carbon, so instead of having a hydroxyl group there, we've been deoxified and we have just the proton there instead, well, that's the deoxification that leads to deoxyribonucleic acid. That's going to be our deoxynucleotide, and with that nucleotide, we would be stringing together deoxyribonucleic acid, or DNA. So if you have 2 hydroxyls, that's for RNA. If you have 1 hydroxyl, that's for DNA. The D stands for deoxified. There are 4 nitrogenous bases, 4 nitrogenous base groups for both DNA and RNA, and they break down into 2 subcategories. We have the pyrimidines, which are made up of a single ring right here. And the pyrimidines for DNA are cytosine and thymine. And the pyrimidines for RNA are cytosine and uracil. No thymine in RNA. Uracil takes its place. The purines, interestingly, that's the small word, but the larger structure because it's the double ring. The purines are adenine and guanine, A and G, and that's the case for DNA or RNA. So we've got 2 families of nitrogenous bases, pyrimidines, which are small, and purines, which are big, and the purines are adenine and guanine. The pyrimidines are cytosine and uracil for RNA and cytosine and thymine for DNA. In addition to being monomers for making RNA and DNA, nucleotides, or at least the nucleotide di- and triphosphates, are also the basis of chemical energy in the cell. When we talk about sugars and fatty acids storing energy, we mean just that, storing energy. The tangible, usable currency of energy in any cell are nucleotides, and predominantly adenosine triphosphate, right, ATP. Here's AMP, or at least an MP, a monophosphate. Not a lot of energy there. But then if we go to a diphosphate, 2 phosphates together, we've got some good potential energy there. And if we go to a triphosphate, 3 phosphates linked together, oh boy, hold on, because that is a ton of potential energy. And we should be able to deduce that using our logic, common sense. Because what do like charges do? Like charges repel. Right? Opposites attract. You get a negative magnet and a positive magnet, they're going to stick together. But like charges repel. You put the positive faces of 2 magnets together and they will push off of each other. So when you get 2 phosphates together in close proximity like this, held together by a covalent bond, there's an enormous amount of potential energy there, an enormous amount of energy of want. These 2 phosphates want to push off of each other. And now if you get a triphosphate, you got 3 negative charges all close together. Oh man, those phosphates are miserable. They're dying to get away from each other. And that energy of want, that repulsion that can't occur, is all the trapped energy of the potential energy of these bonds here. Cut this bond, and that phosphate's going to go flying off, converting that potential energy to kinetic energy. Cut this bond right here, again, phosphate's going to go flying off. The reason why monophosphates contain very little potential energy is because, well, that's just this one negative phosphate, like hanging out there. Okay, I'm negative, but I'm not really trying to get away from anything else, right? So not a lot of potential energy in a monophosphate. And again, when we talk about ATP, the energy currents of the cell, the energy molecule of the cell, everything runs on ATP, got to make ATP. Cellular respirations for making ATP. Oh my God, I ran out of ATP. What do? That ATP stands for adenosine triphosphate. It's the triphosphates that contain the energy. The adenosine is secondary. It's the triphosphates where the potential energy is stored. And so sugars and fatty acids, good for long-term and short-term energy storage, but it's the triphosphates that is the usable, on-demand, present right now energy that our cells are running on. And again, I have the text there reiterating that when you have multiple negative charges bound so closely together, you've got this repulsive energy of want, this potential energy, and then you can release that potential energy as kinetic as soon as you start breaking those bonds. How do you jam on a second and third phosphate onto an adenosine in the first place, right? If those negative charges of those phosphates want to repel so much, how do you actually get the energy in to get them so close together so you can get them to stick with a covalent bond? Well, the short answer of that is that's the story of aerobic respiration. What we do with glucose to make ATP is all about harvesting the energy to make that triphosphate. And we'll talk about that in week 6, I think. And then once you've made these triphosphates, you've got all that potential energy stored in those bonds. When you need kinetic energy, when you need heat, when you need to power some kind of cellular process, you just do a hydrolysis reaction. You break these bonds that are holding these phosphates together, and all that potential energy immediately converts to kinetic, and you can use that energy to power whatever the cell needs powered. And so we see that in this nice diagram from the textbook. Here is the triphosphate. And the triphosphate with all the potential energy of repulsion trapped in these bonds. You can hydrolyze these bonds, break these bonds, and release that energy for intracellular work. But when you break that bond with a water, when you hydrolyze that molecule, well, now you're left with a diphosphate. Well, to take a third phosphate and jam it on, repulsive force and all, jam it onto these two phosphates that are already there, that's going to take a lot of energy to do that reaction, and that energy is coming from the food that we eat if we're a heterotroph, or it's coming from photosynthesis if we're an autotroph. So as cool as all that might be, for me, a molecular geneticist, the most impressive role that nucleotides have is their role in nucleic acids. In other words, their role in storing information. DNA and RNA is a language, make no mistake about it. It conforms to and follows all of the native rules of any other language we have found on this planet. And that language with an alphabet of 4, 2 purines and 2 pyrimidines, encodes information just as all languages do. The language of nucleic acid is the language of protein production. The information that's contained in our genome and the RNAs that are copied from it is the information required for making new proteins. But as far as nucleic acids go, they are storers of information. The nucleic acids that are, well, I should say the nucleotides that are linked together make up a code, and that code can be decoded and deduced by the cell and used as an instruction manual for making proteins. Talk a little bit about that in Bio 107, and you'll talk about that a whole lot more when you take genetics in your 3rd year, probably next semester, I think, for many of you. So we can take these monomers, we can take these nucleotides and link them together by, say it with me now because I'm sick of saying it, by a condensation reaction, right? We can take a hydrogen from one and a hydroxyl group from the other and make water. And with the leftover unpaired electrons, we can create a phosphodiester bond between those two nucleotides. And we can take 2 monomers of nucleotides and link them together into a chain of 2. And then we can take a 3rd nucleotide and link it in to make a chain of 3, and so on and so on. When we do that again and again and again, creating these very long strands of DNA or RNA, these long polymers of nucleic acid, it is not so much the phosphates and the sugars that have anything to do with the information. It is the sequence that has to hold that information. It is the sequence of those nucleotides from the 5' end to the 3' end. It is GATC that contains the information for building proteins. Long words and sentences of nucleic acids encoded in the genome that can be decoded for translation for protein synthesis. So DNA is our long-term our long-term information storage. DNA is both very, very stable as a molecule, and it is privileged in the nucleus where it's safe from any kind of damage or any kind of hydrolysis. And then RNA is our short-term information storage. When we want to make a Xerox copy of a DNA gene, well, we're going to make that Xerox copy in an RNA form. RNA is very tiny, it's modal, it can get out of the nucleus quite easily, it can be used by the cell easily, and then it breaks down easily, and we're done with it anyway, so who cares? So DNA and RNA differ in 3 main ways. The first we've already covered. DNA is deoxified. It does not have a hydroxyl group on its 2' carbon. It has a proton instead. And in fact, that loss of that reactive oxygen is one of the main reasons why DNA is a much more stable molecule, because it doesn't have a hydroxyl group in that second position. Also, as we've already alluded to, the two pyrimidines of DNA are cytosine and thymine, and the two pyrimidines of RNA are cytosine and uracil. They do their own role, or I should say they substitute for one another in those two different nucleic acids. What thymine is encoding in DNA, uracil encodes in RNA, and vice versa as well. They take each other's place in those two molecules. Another reason why DNA is so stable is because it always has a partner, a little buddy strand that's complementary to it, an antiparallel. DNA is nearly always double-stranded, and those two strands base pair together and stabilize one another. RNA is typically a single-stranded molecule. It can sometimes fold into three-dimensional shapes by self-base pairing. Bases of the one strand of RNA can oftentimes base pair with other similar regions, but it's not double-stranded the way DNA is. And as I've already said, DNA is for that long-term information storage. DNA is very static, very boring, just sits there in the nucleus, but it holds the information, the instructions for all the proteins that cell would need. And then RNA is this short-lived, transient, portable, smaller copy. Okay, so those are the four macromolecules, right? We've got sugars, fatty acids as our representative group of lipids, proteins, and nucleic acids, all polymers of their individual monomers. These four classes make up the vast majority of all the chemicals or molecules in any living cell. As I told you at the start, all cells are about 70% water, right? 70% water. We took the ocean with us. The remaining 30% of cells is made up of molecules and chemicals. Of that, about 80% are polysaccharides, sugars, proteins, RNA and DNA, and lipids. 80% of the non-water stuff in any cell falls into one of those four categories. And the remaining 20%, small molecules, inorganic ions, and things like that. So it is these molecules, these four classes of macromolecules, that make life possible on planet Earth. And all of these molecules consist of monomers, monosaccharides, amino acids, fatty acids, and other lipids that we didn't talk about, and nucleotides. Those are the monomers, linked together into polymers, polysaccharides, things like triacylglycerides and phospholipids, proteins, peptides, and nucleic acids. And what is doing the linking, the way that those covalent bonds that are responsible for the linking are made, is through condensation reactions where water leaves as a leaving group and the remaining unpaired electrons come together to create a new covalent bond. This is how you make polymers. This is how you make I don't care if these green boxes are amino acids, monosaccharides, nucleotides, or lipids. This is how you make a macromolecule. You have some existing chain. You want to add a new monomer to the chain. You're going to remove water in your condensation reaction, and the unpaired electron here and the unpaired electron here are going to pair up and make a new covalent bond, joining this fourth subunit to the chain, making that polymer longer. The details differ, right? The bond names differ. Glycosidic bonds for sugars, phosphodiester bonds for nucleic acids, peptide bonds for proteins. Those names differ. The details differ. What the functional groups are differ. But the core themes are the same for all of these. Whether you're making a polysaccharide, a protein, or a nucleic acid, they're all following these same general principles. All polymers are built sequentially, one monomer at a time. The sequence is critically important for all of them, and their synthesis, their creation, must be controlled and regulated and overseen by the cell. Of the four classes, again, it pains me to say, but of the four classes, you could make the argument that the proteins are the most important because they're responsible for catalysis, for regulation, for sensing, for facilitating and executing all of the reactions of life. But it's equally important to note that life would not be possible without any of these. So life couldn't exist without sugars. Life couldn't exist without nucleic acids. Life couldn't exist without lipids. And life couldn't exist without proteins. Proteins do the most, but each one of these macromolecules is essential for life, and life is only possible because we have all four. Okay, so I just want to close out with some returning to non-covalent bonds, because non-covalent bonds play a key role in some of what we talked about today, and they're going to loom quite large in next week's material. So the real question becomes, if structure equals function, if the function that these macromolecules have, they do have because of their three-dimensional structure, what holds that three-dimensional structure in place? We understand that it's the covalent bonds that hold the monomers together into a chain, but what holds the polymer into its three-dimensional shape? And the short answer to that question, which brings us full circle for what we were talking about even last time in our last video, is non-covalent bonds. Non-covalent bonds hold these macromolecules into their three-dimensional shape. Hydrogen bonds, hydrophobic, quote-unquote, interactions driven by water, van der Waals interactions, induced dipole-dipole interactions, and ionic bonds. These four classes, right, hydrogen bonds, hydrophobic interactions, van der Waals, and ionic, these four non-covalent bonds are responsible for holding RNA, DNA, protein, lipids, and sugars into their three-dimensional shapes. And those three-dimensional shapes is then what gives those macromolecules their function. Yes, covalent bonds hold the monomers together into a primary sequence, but it's non-covalent bonds that hold those polymers together into a three-dimensional shape. We call those three-dimensional shapes conformations, and those conformations is what gives those molecules their function. And again, also to get back to echoing some things from previous material we've covered, each one of these non-covalent bonds is quite weak and easily broken. The reason why a protein or an RNA molecule or a sugar can be held into a very specific three-dimensional shape is because of the sum total force of many, many non-covalent bonds. Again, this idea of Velcro, this idea of specificity, this idea of the sum total of many weak interactions gives a very strong interaction. Protein subunits, sugars and the proteins that bind to them, signaling molecules and protein receptors on the surface of the cell. Anytime two different macromolecules need to interact with each other with any kind of specificity, with any kind of recognition, that recognition is occurring through a suite or a host of non-covalent bonds. Where this molecule is negative, this molecule is positive. Where this molecule is positive, this molecule is negative. This is a complementary surface for interaction. Everywhere there is the potential for a non-covalent bond in the light green molecule, there is the matching and opposite potential for that same non-covalent bond in the dark green molecule. And these two molecules come together and fit perfectly. And when they fit perfectly, all those non-covalent bonds come to be, each one of them being quite weak, but in their sum total being very, very strong. And only these two molecules will interact this tightly because only these two molecules fit one another and complement one another in just this way. That's something we've talked about already, and it's something we'll talk about again next week as we go deeper and deeper into protein structure and function. And so to summarize what we've talked about today, life is organic. Life is built on carbon-based organic molecules that exist in an aqueous environment. Carbon is the universal connector atom, and life is built on carbon because all of these atoms need to connect in a specific and spatial way. Cells contain four major macromolecules: our sugars, our lipids, our proteins, and our nucleic acids. Each of them are monomers linked together to form polymers. Sugars serve as energy sources, which again we'll get to much later on in the course, and structural components, things like chitin, things like cellulose. The exterior of so much life on this planet is a sugar exterior, a structural sugar exterior. And these are sugars that are polymers, individual monosaccharides linked together by glycosidic bonds. And then we also have starch and glycogen also as polymers, also as complex carbohydrates, but not for structure, but for energy storage, starch in plants and glycogen in animals like us. Our second class after sugars, fatty acids. Fatty acids are characterized by being hydrophobic. They belong to the lipid class. They're key to energy storage as well. In fact, they have 6 times as much potential energy stored in their bonds, pound for pound, as sugars do. But because of their amphipathic nature, having very hydrophobic tails and very hydrophilic head groups, it is fatty acids and more specifically phospholipids that spontaneously create bilayers, the bilayers that form the basis of the cell membrane. Proteins, the most important, proteins do everything, and they are polymers of amino acids. Amino acids held together by peptide bonds created by condensation reactions, and proteins are the workhorses of life, where their structure directly determines their function. And then we wrapped up with nucleotides. Nucleotides are the monomers or building blocks of the nucleic acids, both DNA and RNA. If that nucleotide contains one hydroxyl group, it has been deoxified. It is the subunit for DNA. If a nucleotide contains two hydroxyl groups, it is a ribonucleic acid for RNA. And yes, these triphosphates are critically important for energy storage because of all that trapped potential energy in those close proximity negative phosphate groups. But more importantly, it's DNA and RNA that contain genetic information in their sequence of bases. The sequence of their purines and pyrimidines is a language, and that's the language for how to build proteins. All polymers, regardless of what specific macromolecule group they belong to, are formed via condensation reactions where water is removed and the remaining unpaired electrons are used to link these monomers together into a specific configuration and sequence through covalent bonds. And then once you have that primary structure of the monomers linked together by covalent bonds, non-covalent bonds will then be used to hold those polymers into a three-dimensional shape and give them a three-dimensional function. And those non-covalent bonds are, again, hydrogen bonds due to electronegativity, ionic bonds due to the full charge, negative and positive charges between ions, induced dipole interactions, also known as van der Waals, and the mutual enemy of my enemy is my friend, water-driven hydrophobic interactions. The shape, and now we'll use the real word, the conformation of each macromolecule is essential for its function. Structure equals function, and that structure or conformation is held in place by the non-covalent bonds we just mentioned. Proteins are the most central to cellular functions. They are the catalysts, the regulators, the facilitators of all of life's reaction, and the sequence and structure of all of these macromolecules must be tightly regulated for the cell to achieve homeostasis, to work optimally, and to use energy and other resources to the best of its ability. And in many ways, the rest of this course is really fundamentally comes back to that regulation and use of these macromolecules. These fundamental macromolecules and the interactions between them, specific interactions given by these interaction faces, collectively enable the complexity of life on Earth. And so that's the end of lecture 3 and the end of the week 2 material. So as you know from the sequence or the format of the course, we will cover any questions from this video that you have in class on Wednesday. And then Friday, we'll just have an open little short review session, Q&A, to make sure you're comfortable with this material. And then the bulk of Friday will be for a quiz on this material as well. So this marks the end of the week 2 material that you'll be responsible for on the quiz. And then after we do take that quiz and we move on to next week, we'll go into proteins and enzymes, and things start to get really, really interesting. So until then, thanks so much for your attention and for watching, and I'll see you in class real soon.